Good evening, participants. So, welcome to the 15th session on macromolecular structure analysis, that is on gene prediction and concept domain analysis. So, we're not just going to go for gene prediction alone. Of course, we have to see what is genome and how the genes are organized in genome and how the genes can be predicted. So, and also we discuss about conserved domain analysis, especially in proteins, how we determine the conserved domains. So, what is the web resource available in what is the database available in NCBI web resource? So, all these details will be discussing on today. So, this is the final session of macromolecular structure analysis. So, before going to this 15th day session on gene prediction and conserved domain analysis, can we recap the last session contents? So, what exactly did we discuss in the last session? So, what are the major topics did we discuss? Do you want any kind of trigger? Like, can I give you some kind of hint? So, what exactly did we discuss? So, we had a cap, so you forgot, is it? So, actually, we are discussing something related to protein structure. After prediction, visualization, we have gone for validation, Ramachandran plot, isn't it? So I teach you how to use Discovery Studio Visualizer. You remember the softwares, like how to use Discovery Studio Visualizer to visualize the protein molecules, how to predict the 3D structure of chemical molecules, how to view the structure of chemical molecules. Yes, good. So designing the structure of a molecule using ACD Chem sketch. Okay, more of hands-on sessions we have seen in the last session. Okay, we have reduced the theory sessions and we have seen maximum practical sessions on last class. Okay, so now today we'll be discussing. So if you see the macromolecule structures, okay, so next to proteins, the macromolecules or DNA, RNA, nucleic acids, isn't it? So DNA, RNA, and so one should know even how a genome looks like, and how about the domain analysis we can predict. Okay, all these things we'll be discussing it now. Okay, let us start with today's session contents. That is on gene prediction. In computational biology, gene prediction or gene finding. I just first check the volume. Is it sufficient for you? Yeah. So gene prediction in computational biology is also called as gene finding, which refers to the process of identifying the regions of genomic DNA that encode genes. Okay. So one should know what is genomic DNA, what is genome, and how the DNA is the genes information, that is DNA is comprising the genome. For this, we take an example like human genome. If you take a human genome, we should know at least one genome thoroughly so that we can compare our human genome with even other genomes. Like, for example, if you're working on mice, if you're working on rats, okay, if you're a veterinarian, you have to think about to work on dogs, cattle, etc., isn't it? So even if you want to study this kind of a disease information of particular animals, one should first know the information of humans. Then we can compare, and if you understand, then it will be easier for you, isn't it? So first, we take human genome as a standard, and we can discuss more about that and how the genes are organized in that. Okay, so first they are mentioning like in computational biology, we generally predict the genes only from the genomic DNA. If so, if you come across genomic DNA, you have two regions, okay, majorly two regions. One is exon. The exon also we call it as coding regions. So these are coding regions, and introns are non-coding regions. Okay, codons. You have, if you see the types of genes, you have coding genes or non-coding genes. When we call as a coding genes, if genes code for protein, then we call that as coding genes. Okay, that we call it as exons. And if genes won't code for any proteins, we call that as intron. Okay, so if for both exon and introns together, we call it as genes. Okay, and if you see this kind of other regions, okay, here starting of the 5' region of the genomic DNA, you have untranslated region, and even at the end, you can at the end of 3', you also have an untranslated region, which we call it as UTR. Okay, in between that, you have number of exons. So according to this, this is exon 1. According to this genomic DNA, we have exon 2 and exon 3. If this is exon 1, okay, similarly here, whatever you have, this is the intron 1 and intron 2. If you have studied molecular biology, you have come across one concept called molecular splicing. Splicing. What do you mean by molecular splicing? So, if non-codon genes are spliced off from the genomic DNA, then we call that as molecular splicing. Okay. So, I told you like even before when I gone for motif prediction, I told you a software for predicting a single motif, and also given one more example of software for predicting multiple sequences motif. Do you remember what examples I have provided for motif finding? Because one should know that even to go for concept domain analysis. So this is my question now. What is the software I provided for predicting motif in a single protein sequence? Give me an example. I have provided a hands-on session in that software. There is an online software. What is that? You remember? Yeah, if you have a cap, well, forgetting. Yeah, what is the software I provided you with hands-on demo for predicting a motif? What do you mean by motif? A motif for of many types like structural motif, sequence motif, and functional motif. These are the types of motif. And if you compare motif, okay, so if you see the types of motif, I told you one is structural, sequence. Very good. Ketan has tried well. Good, excellent. But that is not the correct one. So whatever you mentioned like MEME, that is for predicting multiple motif. I mean motif in multiple protein sequences. If you have three to very good. I got answer from Shreyazi. The answer is perfect. Scan ProSite. So can I get your institution names, Kirtan and Shreyazi? Can you type the your institution names in the chat box? Glad to see that at least few participants are answering the correct answers. I appreciate your performance. Okay, types of motif. We have structural motif, sequence motif, functional motif, and comparative motif. Comparative motif. Okay, comparative spelling is wrong. Yeah, comparative motif. So these are the types of motifs we have discussed. Okay, so based on the protein sequences, okay, we have to go with any of this. Generally, the comparative alone, if you want to compare with multiple, you go with this. But generally, we have these are the types of motif. Okay, so if you go with a single protein, so in XPassive Observer, we have a tool called Scan ProSite in XPassive Observer. Okay, and MEME is a separate software suite. In that package, you have a program called MEME itself to work on uh, predicting motif for multiple protein sequences. But so why I'm mentioning you, why I'm recapping you that thing. So like if you see the important part, the functional part in the genomic DNA. The functional part is what we call the as exons, which we call it as coding genes. Okay, but the non-functional part, that is which is not going to the genes which are not going to translate to proteins. We call that as introns, isn't it? So these introns alone will be spliced off. So during that motif demo, I also told you, may exon 1 and exon 2 can form a gene 1. And exon 1, exon 2, and exon 3 can form gene 2. It depends. Okay. So, like based on the splicing and how many genes you uh, join it, okay, after removing the introns, that based on that you can find the number of genes produced from the genomic DNA. Okay. So the number of whatever I am telling is a gene is default a coding gene. Okay. So the coding gene defaultly we call it as exons and non-coding genes. Once again, I'm telling you, sorry, introns. Okay. Now go to the gene prediction. In its earliest days, gene finding was based on painstaking experimentation on living cells and animals. Statistical analysis of the rates of, if you see the genes, we have types of genes, orthologs, paralogs, homologs, xenologs. So there are types of genes which we have to check. Okay. So based on that, even the gene prediction can be done. If you see the gene prediction, we generally go for a closely related to the target. That is, if you work on DNA binding proteins, which we call it as transcription factors, you want to focus on transcription factors. You won't go to the any other protein domains. Okay, so the DNA binding proteins have a transcription factor domain. So you have to focus only on that, and you have to find any alterations or there in the specific binding sites of the genome or not. Okay, so the DNA binding proteins or family of proteins. Similarly, you can go for hemoglobins, immunoglobulin. Okay, likewise, we can check. There are so many 20 types of immunoglobulin molecules are present, subunits are available. So you can also compare and you can check the specific, you can locate the binding sites of the specific proteins in the genome. Many aspects of structural gene production are based on current understanding of underlying biochemical process in the cell, such as gene transcription, translation, protein-protein interactions, and regulation process. Okay, so which can help us to study in various research fields like transcriptomics. If you are studying about gene transcription, what do you mean by gene transcription? The process of gene transcription is converting DNA molecule to RNA molecule, isn't it? So if you're studying about RNA molecule generating from DNA, that we call it as transcriptomics. And translation, the RNA is going to translate to a protein. So not only you're just studying about a single protein, you study a family of proteins or a cluster of proteins or even protein-protein interactions in the field called proteomics. Okay, in proteomics, you have plenty of these information, translation, protein-protein interactions, whatever I told, and regulation process, gene regulation, gene interaction networks is different, gene regulation networks is different. Okay, so if based on the gene regulation, you can find the metabolites are formed. Okay, so based on the genes, so then one should know about what gene is regulating what gene in this kind of studies, metabolomic studies, and even some kind of structural and functional genomics. Okay, so these are also important to study about the evolution and the conserved domain analysis, etc. Okay, now we can go to a first software called Glimmer. So Glimmer, generally we call that as gene locator interpolated Markov model, yeah, which is used to find genes in prokaryotic DNA. It is effective at finding genes in bacteria, archaeoviruses, typically finding 90 to 99%. If you see the codons, that is coding genes, okay, especially exons, okay, the exons in yeast is maximum. The percentage of coding genes in yeast is more than 99 percentage. So exons level is more than 99 percentage in yeast because the so only the protein you can find large protein-protein interaction networks in the yeast, and that is the first kind of a species that they explode a maximum kind of proteins are interacting with each other in multiple pathways. Okay. And if you see humans, we have less than 2% of exons. So only less than 2% of the total genes in humans code for proteins. That means more than 98% of the genes won't code. So it means these are, we call it as introns. Okay. So we have, you find a variation. Okay. Even in E. coli, you can find nearly 98 to 99% code for proteins. But that is not the case in mammals like human. So one should know even more about the details of the exons. So Glimmer was the first system that used the interpolated Markov model to identify coding regions. The Glimmer software is open source and is maintained by Steven Salzberg, Art Delcher, and their colleagues in John Hopkins University. The original Glimmer algorithms and software were designed by Art Delcher, Simon Kasif, and Steven Salzberg. So the URL for downloading Glimmer. So you can download the freeware, okay, Glimmer in offline, and you can work on that. So before going to that software, so try to understand the difference between the two different gene structures. That is gene structure of prokaryotes from the eukaryotes. So if you are studying protein engineering, okay, or protein science as a subject, okay, you might have seen this. These are some of the fundamentals one should understand. So that is in prokaryotic genomes. Okay. Initially, if you see this kind of genomic DNA, you find a promoter for a regulator gene at the beginning region. I was showing you an overall gene 5 prime to 3 prime end. But what is there in the structure of the gene? So that's what I'm exploring here now. Okay, so the promoter for the regulatory gene you can find initially. Next to that, you can find the regulatory gene. And next to that, you have a promoter for lac operon. Lac is a lactose operon. Okay, so and if you see after the promoter, you can find the operator and the structural elements of the gene, that is Z, Y, and A. Each and every part of the structural gene code for a particular proteins. That is, for example, this is a structural gene for beta galactosides. And Y is the structural gene for beta galactosidase permease, an enzyme. And A is the structural gene for beta galactosidase, beta galactoside transacetylase. Here, this is beta galactosidase. This is another enzyme. So, three different enzymes you can find. So again, I'm telling you, in prokaryotic genome, we start with a promoter of a regulator gene, followed by regulator gene, and promoter for lac operon, so lactose operon, and then immediately followed by operator. After operator, you can find the structural genes. There are three structural genes, each structural gene code for an enzyme. The first is a gene code for beta galactidase, and Y code for beta galactoside permease, and the A code for beta galactoside transacetylase. Then we can go for so the entire up to this promoter to the structural region. That's what we call it as lac operon. Okay, so this is the concept we find in prokaryotic gene structure. Okay, if this is these are the elements found in prokaryotic gene structure, how about eukaryotic gene structure? In eukaryotic gene structure, we have so more about this. We can study in molecular biology. Okay, so we have Primrose, Twyman. It's a very thick book. Okay, nearly 700 to 800 pages. We have more details on this to study. Okay, so even in protein, you might have seen how to engineer these kind of genes. One should know even how to go for genetic engineering. Okay, so like if you want to go for pro-insulin manufacture from E. coli, one should understand the prokaryotic gene structure, isn't it? Where we can incorporate that target DNA. Okay, so likewise, this is an example for a lactose, a lac operon. So likewise, we have different structures. So most of the genes have this kind of a structure. Now, let me take you to the general eukaryotic gene structure. If you see the general eukaryotic gene structure, initially you have the enhancer. This we call it as a distal element, and the promoter is a proximal element, and a tata over box. Okay, followed by exons and introns. So whatever I mentioned, exons as a coding genes and introns as a non-coding genes. I told you in mammals, okay, especially which comes under eukaryotes, you find maximum of introns, that is 98% and more. I explained you know. So whatever I explained, so you can find this in these regions. Okay. So you have an enhancer distal element at the beginning, promoter proximal, followed by data box, and these genes, exons and introns, coding and non-coding, later connect with the enhancer of distal element. Okay. So likewise, you can connect with again. Similarly, like based on the genes, you can find the extension of the genes. Now, let me take you to the genome because when we study for the gene finding, they're telling that we are going to predict genes from the genomes. So one should understand what's there in the genome and how the genes are going to be predicted from that. So for this, we can see NCBA. This, of course, we have a genome database. I told you instead of protein, if you select genome and if you type a species name or if you type a protein name, obviously that will take you to the NCB genome website and tell you how about the protein is going to code, in which chromosome it's going to code, all this information you get. Okay, so related to the NCBA genome, if you go to this URL, that is in NCBA datasets genome, okay, so you can find the total number of genomes deposited in NCBA. So totally, if you see, 24.2 lakh 2 lakh genomes are deposited. Okay, out of that, the reference genomes are 36,070 something like that, 36.75K, and annotated is around 19.9 lakh genomes are deposited. So you can find these species majorly, that is eukaryotes, archaea, bacteria, and viruses. So whatever genomes they discovered, okay, or in this kingdom, in this taxonomy, okay. So if you go to the eukaryotes, you can find our human genome information. So if you want to search genome information, you can use various database identifiers like taxonomic ID. Okay, eukaryotes have a taxonomy ID, and if you go for with the specific species, okay, you will find a kind of a uh, name or ID. You can provide that as an identifier, or you can give an assembly name. You know, the genes are assembled to form a genome. So there will be a kind of an assembly name for that. That is after the during the sequencing, okay, especially through the Illumina or any other techniques, ion torrent, etc. So whatever the contigs are the part of the DNA fragments which they use it for construction of genome. So for during construction, you have an assembly name. So even you can give assembly name as the identifier for your project, for your sample, WGS, nucleotide accession number, etc. So just you have to remember at up to present 24.2 lakh structures are available. I mean to 2 lakh million, 2.42 million genomes are available. What is genome? How a genome looks like? Do you know? What is the shape of a genome? The shape of a genome is circular in shape. Okay, this is not spherical. This is just a circular, like a single D information. But how this genome is information we get? So take an example of NCBA, I mean human genome. In human genome, the genes, whatever we take, are only from the chromosomes. So generally, if you see the experiments, how they go for sequencing the genome, they take a drop of a blood, okay, and just the drop of a blood can have around 14 to 15 cells. So these cells generally they just burst, okay, in a bubble. If they burst, you can find dispersing of chromosomes and can go for karyotyping to distinguish what type of chromosomes you have. But whatever chromosomes you take, okay, so initially if you see the human, we have 22 haploid chromosomes. I mean, 22 haploid autosomes. There are two types of chromosomes, isn't it? One is autosomes and the other is sex chromosome. Autosome, if you see, we have 22 haploids, and if you see the sex, you have X comma Y. So totally, if you see the haploid chromosomes alone, which can map a circular genome, you find 22 plus X and Y. So totally, you have 24. So these 24 chromosomes give this circular shape. But there is also one more information, that is mitochondria. So for whatever genes you find from the mitochondria is going to give another kind of a genome, that is mitochondrial genome. Again, it's a circular shape. You got it? So autosomes and sex chromosomes together, you find this. So you see here, you have first two chromosomes, which is the largest chromosome, start from here. Like how you drop the information in the CD or DVD. Okay, you have a center thing which you insert in the CD player or DVD player, and if you try to write, obviously the data is populated. You find the gray color. Wherever you find the gray color, you can tell that the data is there in the DVD, isn't it? Similarly, like a DVD, the genome looks like this. The first genome information you can find from here to here. I mean, first chromosome information you can find here to here. And this is the second chromosome and third chromosome. Likewise, you can see all the 22 autosomes are there, and X and Y. Okay, X is of course a large chromosome. You can find more information on that later. Y. So this is one such genome, and mitochondrial genes you can get in another gene. Okay, you got it. So this is what the concept they used for in NCBI genome. Now let me take you to the gene prediction methods. So what are the different gene prediction methods we have? Protein coding genes. We can able to predict using ab initio homology based and non-protein coding genes. We can go for tRNA, that is transfer RNA, ribosomal RNA, and small. You have microRNA, small nuclear RNA, sRNA, siRNA. There are types of RNA. So all these things comes under non-protein coding genes. Okay, so these are the prediction methods we generally use. So what are the Markov model based tools available? So Markov model based, we can go with gene mark S, glimmer, RAST, AMI gene, EC gene. Okay, so these are the prokaryotic gene finding methods available, which are developed under Markov model algorithms. So in Google, if you default type gene markers or glimmer, it takes you to that website where you can go and predict the genes information. Okay, so gene finder tools. What are the different tools available, and what is it for developed, and what are the different output file formats we can get? So the first we see Prodigal. The Prodigal is developed for bacteria and archaea. The output file formats are GPK, GFF, SCO. And the second what we see is the gene mark S. The gene mark S is used for is developed for prokaryotes. Output file formats are algorithm specific. And the third tool is Raster, that is developed for bacteria and archaea. And the output file formats are GTF, GFF3, GenBank, and the EMBL. The fourth tool is Glimmer 3, which is for developed for prokaryotes, algorithm specific. And EC gene is for prokaryotes, EFF3 is the output file format, and AMI gene is for prokaryotes, EMBL, GenBank, and GFF or the file format. So one can try with these kind of a tools for these species, for this kingdom of organisms, and these are the output file formats we can get from these tools. So one such example is Gene Primper. It is a computational evidence-based post-processing pipeline that identifies erroneously predicted genes. The list of gene anomalies reported, so short genes, long genes, broken genes, interrupted genes, unique and dubious genes. So how this is actually predicted, what is the kind of data flow? So if you see the gene primp data flow, okay, initially you take the genomic DNA, You take it to this kind of a softwares like Prodigal, GeneMark, RAS, Critica, Glimmer, Imagine, etc. Then you will be getting a gene that the gene calls, and you send it to the gene prim. So you get a report of these distinguished kind of genes information: short, long, broken, interrupted, unique, dubious. Whatever genes information we get from the sequencers. Okay, if you see in next generation sequencing, generally they call that as reads. Whatever information, the genes information they get, a large gene information, they call it as a reads, READS. Okay, so according to that reads information, okay, so if you find high quality reads, then it will be easier for them to sequence. I mean, to assemble, to form. Okay, if you're having a low quality reads, that is of not significant. So one should also go with statistical analysis with the next generation sequencing to find how much high quality reads and low quality reads are produced from the sequences. So obviously one should know about genome assembly during the construction of genome. First we should get a genome. I mean, first we should get a gene, and then we have to gene is a general term, but during sequencing, we have contigs, that is the small fragments, which can be even generated after the shotgun sequencing. So after shotgun sequencing, whatever you get like contigs, you try to assemble it using the genome assembly software and try to map that in a circular genome. Not only human genome is circular in shape, even other species genomes, whatever I told, 2.32 genomes available in NCB, all the genomes are in circular in shape. They are mapped according. Only they have taken the chromosome genes, both exons and introns. If it is an eukaryote, okay, they have taken the genes and they just mapped it in the form of a circular shape. Okay, so genes prediction is one of the step for the genome construction. And even if you want to find what is the gene responsible for a particular disease, can be a rare disease, cancer, etc. So one should know about the major gene. So to know about the genes information, you can also search that in the already constructed gene. Okay. So gene print. So we just seen the data flow, isn't it? But this much information is there in the flow chart. So gene print works with multiple files. Okay, so many intermediate files are generated and so many tools we have to use in between to get a kind of thing. Okay, so this is a flow chart. We use all the types of blast, especially which can handle the gene, protein. Okay, and we try to find the types of genes. That's what the major output of the gene. The types of genes is putative mid chains, which short and long chains, broken chains, interrupted genes. So all this information we can find from gene. Gene mark. This is a family of gene prediction programs developed by Georgia Institute of Technology. This is another software. In this gene mark, you can predict. You have a tool separately for predicting the gene in bacteria, archaea, metagenomes, and metatranscriptomes. And for eukaryotes, they have a different tools. So one have to click this hyperlink and you can download the tool and you can work with gene finding. Okay. So similarly, you also have for viruses, pages, and plasmids. And the URL for the gene mark is mentioned below. So gene frame just a comparison. Okay, that is even we can compare genes. I told you comparative analysis, comparative motive, isn't it? So similarly, comparative genome analysis can also be done. Okay, so for this here in this case, Mycobacterium species. Okay, uh, we are comparing with the Mycobacterium palustris. So this species is different and this species is different. If you try to check with different gene finding tools like GeneMark, Glimmer3, Prodigal, RAST, Imagine, how many genes you got for this Mycobacterium species? And how about for Mycobacterium palustris? I told you these are the outputs you get, isn't it? Coding sequences, short genes, long genes, missed genes, unique genes, dubious genes, broken genes, interrupted genes, all this information in different tools. Whatever I mentioned, gene marking, multiple prodigal trace damage genes can be found out from this. Okay, this is a comparative kind of gene analysis in the genome. And if you see this kind of a gene prediction, I told you majorly we have two types of genes. So one should distinguish whether to go for exons or introns. The protein coding genes is what exons, and the non-coding genes is what called introns. So you have different types of RNA in that. So one has to first distinguish the thing, and then they have to use the software corresponding. Whatever is shown in the previous slide, here I am showing in the form of a flow chart. Okay, ab initio. Now we can see what are the tools present in ab initio. Gene mark, protocol, EC gene, AMA gene, limit3, RAS temperature. Whatever I mentioned comes under ab initio. For homology based, you can go for the blast HEV2 plat. Okay, so the plat is just like a blast tool, but this is used for genome analysis only. This is not for sequences. If you want to use uh, a search in a similar kind of algorithm like blast to search in genomes, then you can use plat. Okay, so this is based on homology based. You know what is homology? If you have a sequence and if you have multiple sequences in the genome, you just blast with the genome and try to get what is the exact sequence you get. Okay, so you get for a sequence similarity if you have a sequence alone. That's homology based. And for the RNA genes, that is introns, they have these softwares: tRNA scan, SC, RNA sRNA target, sRNA predict. And so the final RNA predictions can give you the final result if it is a non-coding. But if it is a coding gene, you have to try with initially gene prim and then go for manual variation. Okay. So whatever I told you using the basic software comes into gene prediction. And for gene prediction improvement, you go for gene prim, manual variation, and exploring your final result. So this is a basic flow chart of for how to predict the genes from the genome. So conserved domain analysis. Before going to the conserved domain analysis, I can give you some few more information on genomes. Note that. So one should know something about what's happening in the world. I was explaining you about the genome that 22 autosomes and X and Y together gives you the entire human genome in circular shape, isn't it? Can someone tell me what is the size of a human genome? I think someone who have already studied this may think that. What is the size of human genome? We should know at least one genome with all this information. Yeah, during annotation, shall I include mitochondrial gene in reference genome? Oh, no, mitochondrial genes or something different. So if you're working on mitochondrial genes, then you can explore. If it is a normal kind of gene, okay, one should know whether the gene is present in nucleus. So whatever I'm telling, the autosomes, 22 haploid autosomes and the X and Y are present in nucleus, isn't it? So you are bursting the nucleus of the cell to release the 24 chromosomes. But mitochondria is a different organelle. You have different genes composition. If you are specific that you are working on mitochondrial targeting, then you have to check for mitochondrial gene information reference gene. Or else you just focus, I have to focus only on the nucleus chromosomes, that is 22 plus 2. In comparative genomics slide, different tools are showing different number of coding regions. What should I consider? So then you should think about to get from the reference literatures. So for mycobacterium, you get this kind of an information. But how about for human? So definitely you have standard literatures. One should follow the literatures for using that tool for a specific species. Okay, even though they're telling they're using Markov models, as one of the machine learning algorithms. In some tools, in addition to the Markov models, they might have used one or two more additional machine learning techniques. That gives you a better kind of precision. If you are, again, I'm telling you, not only the machine learning algorithms, what they use gives you the precise results, even data information. If you have a large data, okay, that is in a sample, if you have a large data, then whatever that we call it as a training data set. Okay, if you have one genome and if you want to compare with the one genome, you won't get a good result. But if you have 1,000 genomes of a same species, and if a person is suffering from a disease, instead of searching with a single human genome, if he is searching with 1,000 genomes, he may get a kind of precise information of what exact gene uh, he is diagnosed with a particular cancer, and so the treatment will be easier based on gene targeted delivery. So instead of a single genome, one can go with multiple genomes. So in comparative genomics, uh, multiple data as well as how the algorithms are used, that is mostly dependent on how the tools are working and how the tools are showing the exact results. Yeah, just I asked you a question and I'm getting some answers from the participants who never answered, but very good. I got answers from the others also. I'm happy that even the others are interacting with. I just asked you about the size of human genome. Then instead of the coding genes information, that is whatever you mentioned is coding genes in humans, 25,000. So instead of mentioning this, we can give this Pusha's answer. That is 3.2 billion base pairs. This is the exact answer. But if you search in Wikipedia, they might have given 3.1 billion base pairs. Okay, so actually the 3.1 is earlier information. Now, if they're using a proper kind of a sequencing technologies, we can sequence up to 3.2 billion base pairs. In GAT, they have asked a question like, what is the size of the human genome? Okay, they have given choices like 3, 3.2, 3.4. What you can choose, even the Wikipedia they have given 3.1, but now they have increased up to 3.2. So, yeah, if you have 3.1, you select that. But if you instead of 3.1, if they give 3 and 3.2, you have to select 3.2. This is the exact one. Okay, 3.2 into 10 power 9 base pairs. Okay, this is the size of the human genome. Okay, this much genes are encoded. Okay, now let me talk about the 1,000 genome project. This is what for the Dr. Priyanka I was explaining. That is instead of just searching a gene with a single genome, because a single genome is constructed only from a single human, isn't it? If only if you compare 1,000 humans, then you get some kind of reliable information on the particular disease. So the 1,000 genomes taken place from 2008 to 2015. You know, the first human genome was sequenced in the drafted in 2003. They have started 1999. Of course, they're about to publish, but they found very less genes. 55% of project was completed at that time. They got a kind of a good fund. So around, the funding for establishing the first human genome is the cost of the project is equivalent to the cost of Wikipedia. The if you want to search any information, we default use Google as the search engine, isn't it? If you want to get basic information, that is before emerging of AI softwares like ChatGPT, etc., everyone rely on the Wikipedia, isn't it? So the cost of human genome is around $9 billion. Okay, $9 to $11 billion at initial. But now the price is gradually reduced to $1,000. So now even the Indians, okay, are affordable uh, to sequence a entire genome. If he's facing with the rare syndromes or if you rare diseases, genetic diseases, cancer, etc. So like how he is spending for an mRNA X-ray. Similarly, even the doctors ask him to come with the genome sequencing. Just spending $1,000 can provide you a detail of 3.2 billion base pairs information. But whatever, even if you go for sequencing, he has to check the appropriate kind of diagnosis, isn't it? Checking what is a defective gene out of this much basis gene. So one should try with this 1,000 genomes. Okay, so after 2003, in 2015, we got a fully established 1,000 genome project. Okay, so this is generally used for these diseases: cystic fibrosis, Huntington, diabetes, heart diseases. So these are emerging diseases, isn't it? So emerging fatality in Indians and almost everywhere in the world. Heart disease is the first disease. I mean, this ranks number one in percentage of fatality. Next to that, diabetes. In India, it was like that. So one is focusing. So we need an import. We, we got an importance to get to go for 1,000. So even India has a 1,000 project. The consortium aim to discover 95% of the variants. Okay, here's some information. We also have a 1 lakh genome project. This was actually completed by UK government. Okay, that is this was managed by Genomics England. Okay, so it was completed in 2019. Okay, the first release done at the time. And in June 2020, a UK-based biotechnology company announced this they can link this genomic data with academic research institution. So anyone can now even access this 1 lakh genome. If you get a kind of a sequence from the sequencer, one can try with this 1 lakh genomes to find the exact information, okay, exact defect genes, etc. So epigenome, this is something different, isn't it? Genome is different and epigenome is different. So first, one should understand what is epigenetics. Then you can understand what is epigenome. Epigenetics describes a variety of features of the human genome that transcends its primary DNA sequence, such as chromatin packaging, histone modifications, and DNA methylation. So, and which are important in regulating gene expression, genome replication, other cellular process. One should understand whatever we study in post-translational modifications. Okay, like the DNA methylation, etc. So this comes under epigenome. So what genes can go for post-translational modification? That we map it and what we call that as epigenome. Epigenomic, that is epigenetic markers strengthen and weaken transcription of certain genes, but do not affect the actual sequence of DNA nucleotides. The best example they provide is the DNA methylation, which is a major form of epigenetic control over gene expression and one of the most highly studied topics in epigenetics. Okay, now pangenome. What is pangenome? This is again one more technical term. The pangenome is also called as supragenome. Supergenome is different. Supragenome is different. Supragenome or pangenome. This is the entire set of genes from all strains within a clade. What is that clade? Clade of organisms. A similar organisms. Okay. So like Mycobacteria species, you have different species, isn't it? So all these things comes in a single clade. All different strains. Mycobacterium bovis, Mycobacterium tuberculosis, Mycobacterium septicemia. We have different species, isn't it? So we can bring together. And even we have human pangenome. Now, the first human pangenome was constructed from 47 ethnic peoples. Ethnic. So generally, the wrong people use racism, isn't it? So if you see in India, we can classify ethnicity as like North Indian, South Indian. Generally, this is how we can majorly classify, isn't it? So one can see the face and immediately can tell whether he or she is a North Indian or South Indian. So in India, majorly we have two different ethnicity. Similarly, if you take US, you have Native American, Latin American, Indo American, African American, We have different ethnicities there also, isn't it? So if you take all these kind of an ethnic people, okay, in developed countries and developing countries, totally they got 47 ethnic people, ethnic datas. Okay, you understand? So from Indian American, they might have taken only one genome, and African American they might have taken only one genome. Likewise, they've taken 47 genomes to get a kind of a human pangenome. So, what is the purpose actually? The purpose of construction of pangenome is to focus on a specific, a most common disease and even a rare diseases for, and that is the diseases specific to the region. We have heard pandemic, epidemic. You remember these terms? This is generally we use for infectious diseases spread, isn't it? Based on the region, how much region is spread. Similarly, if you see one example, I can give you an example like melanoma cancer. Did you hear about melanoma? What is melanoma? Melanoma is a skin cancer. If you see the melanoma or skin cancer in India, we have less than 0.001 percentage of the people getting infected with skin cancer. Very less when compared to any other countries. You know the reason? The reason why we don't get melanoma, including the Africans, is based on the skin color. We have dark skin colors. So whatever epithelium, whatever skin cells we have, that won't affected by UV radiations. If so, obviously one can able to find that melanoma cancer is not going to affect the Indo-Americans, but it affects the Native Americans. Okay, so instead of going to the pan genome, he can focus on specific thousand genome, something like that. You got it. When we can go for pan genome, the pan genome is something like if you're focusing on lung cancer. The lung cancer is a common spread. Okay, even in USA, many people are dying because of lung cancer, and even in India, this is of course a major cancer. Then, if you're focusing on this kind of a lung cancer, so whatever pan genome is constructed, it will be definitely useful for you. But in this case, like melanoma cancer, you have to focus on a specific people, then ethnic people, then this is not applicable. So one should smartly decide what kind of a genome can be used for comparison. So here you have two types. Okay, one is open pan genome. Equal is an example for open pan genome. Where what is open pan genome? Number of new gene families in taxonomic lineage keeps increasing without appearing to be asymptotic, regardless how many new genes are added to the pan genome. Okay, and if you see the closed pan genome, the example they provide is Staphylococcus ludogenesis. Most of the genomes, pan genomes, are open genomes, open pan genomes. But few specifically, okay, like the Staphylococcus species comes under, which is a commensal bacteria, comes under. That is most of the infecting disease-causing bacteria. Okay, is having a closed pan genome. Okay, and pan genome versus core genome. So if you assemble, okay, so you can find some kind of differences. For this, you can use PPGA software. Okay? That is, if you want to compare these genomes, you can use even this software, PPG. Okay, let me take you to the conserved domain analysis. We have very less time. So I was discussing about conserved domains. The conserved domain database is a database of well-annotated multiple sequence alignment models and derived database search models for ancient domains and full-length proteins. You know, domain. You remember in protein structures, I was explaining you, there are 5 levels of protein structure, and the domains is generally we find in fourth level structure, isn't it? In third level structure, we call that as motif, and fourth level we call that as domain. What is conserved domain? A conserved domain is the uh, part of is a chain. Even you can consider this a protein chain, which is conserved. Like most of the characters, if you take a DNA sequence, whatever characters you present, If you take a protein, whatever amino acid, what is amino acid? Amino acid is nothing but a a three a codon of three nucleic acids, isn't it? AUU can contribute one amino acid. UAA contribute another amino acid. Okay, and likewise, okay, GGG contribute one amino acid. You know, no? So I told you a different codons, nearly 61 codons code for 20 different amino acids. So obviously one can say that if whatever domains you have from one species, from ancestral species, if it is following to the immediate species, so one can find most common domains. Like you can find nearly 1.7 proteins alone. 1.7% of the proteins differ in chimpanzee and humans. Only 1.7 percentage. So it means that more than 98% of the proteins of human and chimpanzee are similar. Okay, so how can find? Of course, even this is one of the analysis, conserved domain database analysis, which uses well annotated multiple sequence alignment models and uses the full length proteins to get a kind of an output. This conserved domain content includes NCPA manually curated. I told you this conserved domain database, okay, is available in NCBA itself. But how this is going to work in NCBA? You know, this NCBA is linked with various external source databases like PFAM, SMART, cluster of orthologous genes, TRK, THR FAMs. So this is going to link with the different databases and you're getting a kind of concept domain information. What is unique about NCBA curated domains is that they use 3D structure information to explicitly define domain boundaries, align blocks, amend alignment details, and provide insights in sequence structure function relationships. Okay, so you have to get a kind of manual curated models. You have to go for clustering, etc. These are some of the steps happening in between. Like even if you go for protein structure modeling using homology modeling, they ask you to simply input a sequence and click develop model, you get a model. But what is happening inside? Nearly 6 to 7 steps are happening inside in virtually, isn't it? Similarly, even in concept domain analysis, these are some of the steps you can commonly observe. So in concept domain analysis, just give this URL. That is in NCBA, you have a structure. The structure database is linked with MMDB. If you go to the NCP and if you type a structure, if you select the structure and give any protein name, you get a 3D structure of a protein, isn't it? So, but instead of a structure, there is also a kind of a URL extended to concept domain database. Okay, if you type this URL, it takes you to this kind of web page where here it asks you to paste the protein or nucleotide query. I told you the concept domains, you can try with both protein or coding nucleotide sequence. You can't give it a non-coding nucleotide sequence. Okay, an mRNA which code for a proteins. Okay, so that kind of a nucleotide query you can paste as an input, or better paste a protein. Click submit. So what is the information you can get? You remember, I have taken 5 sequences of human insulin ligand. I mean, insulin ligand of 5 different species, which I used it for multiple sequence alignment. Okay, 5 different insulin ligand FASTA format sequences. I've already provided you a demo how to go for multiple sequence alignment and how to go for construction of phylogenetic tree. The same FASTA format sequence, if I give that as an input, okay, by pasting the 5 protein sequences, you get an output like this. This is the output of conserved domain analysis. So what output you get? So what is the UniProt ID? So this is the data, and hits by hit type is specific, and position specific scoring matrix ID is provided for this. And so what is the conserved domain? 25 to 108 of TANRI is matching with 26 to 110 amino acid of human, and it is matching with the 26 to 108 amino acid of pig. Okay, and how about bovine? How about CVPO? So the concept of the matching amino acids, what we call it as domain matching. Okay, we are finding using this concept domain analysis, and a bit score is provided. So even if you want to check with human what is close, 133 is close with 129. So I can tell this is one of the entry is having a kind of a close relationship. I think that might be a kind of a monkey type, okay, kind of a chimpanzee type. So and we are getting a close relationship. Okay, so that is related to insulin ligand. So we have so many proteins. You can try with so many proteins, and you can try you can check the concept domain analysis. As I already mentioned, the human and chimpanzee just vary with 1.7%. So we can also find similarly. So any doubts you can ask me at present. I have completed my session on. So one can just go to this URL and just paste the thing, and you can try it out. Okay. So just like this, copy and paste, and just input a sequence. Any doubts you can ask me meanwhile. Just in Google type NCPA consult domains, it take you. So simply you have to take a FASTA format and input. So you know that in desktop have the 5 sequences FASTA format. Where is this? This is the FASTA format of 5 sequences. I simply open with Notepad or Notepad, whatever it is. I just opened a Notepad. Just like that, I'm copying the 5 sequences. So after copying the 5 sequences, you paste it. Okay, submit. You get a kind of an output immediately. Okay, the simple way of how to go for concept analysis. Within 30 seconds, you get an output. Okay, so you got an output like this. Okay, you can record the output. You can download the output in various formats: ASN text, XML, JSON, flash text, etc. You can also have different options for checking the output. Okay, any doubts? If no doubts, thank you very much for patience and interaction. On next session, we'll be discussing on the applications of bioinformatics, that is on system biology and synthetic biology. Later we go for microarray data analysis. Okay, thank you very much. Have a great day to you all.